Today's video is talking about sexual reproduction, which you can find in chapter 9.2 of your textbook. This video will be covering the first 2 dot points here in the study design. How an offspring from 2 parents has a unique genetic identity, and the key events in meiosis that result in the production of gametes from somatic cells, including the significance of crossing over of chromatids between homologous chromosomes in prophase 1 and the non-dividing of the centromere in metaphase 1. So while that sounds like a mouthful, we're going to break it down, and it's actually going to become reasonably simple. And you can always watch this video again. There are lots of good videos on meiosis as well. So first off, let's talk about the 2 parental contributions. In contrast to asexual reproduction, we have to have 2 sets of genes to sexually reproduce. So even in parthenogenesis, it's talking about a faux fertilization. The reason we call it a false fertilization is because you're fertilizing with your own genetic material. So it's not really a fertilization where you can create crossing over. So sexual reproduction involves genetic contributions in the form of gametes from 2 parental sources to their offspring. In animals, gametes are the eggs produced by female parents and the sperm produced by male parents. Gametes are produced in specialized reproductive organs known as the gonads. In females, the gonads are the ovaries. In males, gonads are the testes. The cells in the gonads that give rise to gametes are termed germ cells. So these are 3 that I would like you to put on your cue cards. So if you put in gonads, which will be the reproductive organ, and then you need to put in germ cells. So the gonads are made up of germ cells, which produce gametes, gametes being the 3rd one that you should put on your cue cards. Multicellular organisms are composed of 2 types of cells, somatic cells and germ cells. Somatic cells are cells apart from the sex cell, so that's everything else. That's everything from head to toe, all of our skin, internal organs, everything. And germ cells are the cells that give rise to gametes. So sperm for male and eggs for female. Gametes are formed via meiosis in the gonads. And gametes are the only thing that is formed via meiosis. Everything else is reproduced and maintained via mitosis. So the egg or sperm cells that are produced as a result of meiosis are haploid, which means that the number of chromosomes in the gametes is halved. Normal eukaryotic organisms have diploid cells, which is 1 set of chromosomes from each parent. So you will have a set of chromosomes from your paternal and your maternal line. Paternal being your father, maternal being the mother. So female gametes are large and immobile. They contain food stores needed for the embryo, which is why they're so large and immobile. So women are, in humans, are born with a finite number of eggs, and these are, like we say, large and immobile. So they're not the ones that are going to be finding the male gamete. Male gametes are small and usually have a flagellum attached for motility. So this allows them to move towards the egg. After fertilization, 2 haploid cells fuse to form a diploid zygote, and there's a couple of different biological mechanisms to get them to fuse, which we did watch in another video, and I'm happy to send that link out to you later. So fertilization is the fusion of these 2 gametes to create the zygote. Once it's at that zygote stage, then it's dividing via mitosis to produce a large number of cells which begin to differentiate, which we've talked about in our potency and our gastrulation and things like that. So this process, when we're talking about meiosis, meiosis is only to create the gametes. Everything thereafter is mitotic. Meiosis is the process that produces gametes. It only occurs in eukaryotes and only in gametes. This is the process that creates genetic variation. To maintain the chromosomal number of a species, or how many chromosomes it has, the number of sets in the somatic cells versus the gametes differs. So humans have 23 pairs, or 46 chromosomes, in their somatic cells, and our gametes only have 23 chromosomes, which aren't paired. This way, when the ova and the sperm combine, there'll be a total of 46 chromosomes, and they'll form pairs, and they'll be known as homologous chromosomes. So here, I would like you to put in your cue cards, genetic variation, chromosomal number, and homologous chromosomes. This is a video that we're going to watch in class. It's from Crash Course Biology, where the sex starts, which I think you'll really benefit from, and it really talks about that crossing over process. The next thing we have to talk about is meiosis as a reduction division. So this is how we start to get some genetic variation. Meiosis is a reduction division because it reduces the number of chromosomes to half of that of the somatic cells. Gametes only receive 1/2 of genetic information of the parent. So humans have 22 homologous chromosomes and then 1 set of sex-determinate chromosomes. Sperm have 22 homologous chromosomes and either a Y or an X chromosome to determine the sex in each of 4 sperm. Oogenesis results in only 1 egg and 2 polar bodies due to the uneven split of the cytoplasm. And it also has 22 homologous chromosomes and always an X sex chromosome. So eggs will only contribute the X. So the first half of, if you have a look up here, we've got the 2 X's. The females will only contribute, so the oogenesis, the female gamete production, will contribute this X regardless. It's the sperm that determines the Y or the second X chromosome in our sex chromosomes. So how does this happen? So we start off at metaphase 1. So this should look pretty simple for you guys because you've done something like this before. So we're starting at metaphase 1 because you know prophase is where it all starts to condense, the cellular envelope breaks down, and we're dealing with that kind of stuff. So that's the exact same as mitosis. So here, metaphase, we've still got our microtubules and we've still got our spindle equator and we're lining up. But these are our sister chromatids. So that, see how in anaphase, we're actually pulling not a single chromatid, but a set of chromosomes backwards. So here, we're starting with 4N. So we have a sister chromatid here, a sister chromatid here, and they're lining up alongside each other along the spindle equator. Then here, when we start to pull back, we're taking 2 sets of chromatids backwards with us. We then go into telophase and cytokinesis 1. So this is when it would kind of look like a normal cell that's had its synthesis happen because we've got 2 sets here. And it's the same here, cleavage furrow and cytokinesis just as in mitosis. Then we have to go through it again because what we're looking for is only one of these sister chromatids. So we start at prophase 1 again, our nuclear envelope breaks down and we're moving to either side to create a new spindle equator. This is now our spindle equator. And anaphase 2 looks like a lot like anaphase in mitosis. Then we have our telophase and cytokinesis. So the reason we're doing 1 row here and 1 row here is because we've got these 2 cells. So we start with 1 cell, which has 4N. We've still got 4N. And now, after cytokinesis, we have 2N. But we're looking for 1N. So these 2 cells undergo the second half of meiosis, and they're just labeled prophase 2, metaphase 2, anaphase 2, telophase 2, just to be very clear. And it's our same process again. So I advise you to re-watch that Crash Course Biology video if that's not making so much sense for you yet. So meiosis starts from a somatic cell, and it does involve interphase, prophase, metaphase, anaphase, telophase, and cytokinesis, but twice. So it does begin with one cell, and here's just a list of where you can break it down. And I've also put the ploidy down here for you to kind of keep track of where you're doing that while you're labeling your own diagram, which you should be creating in your notes. So prophase 1, chromosomes condense, composed of 2 chromatids connected at the centromere. The crossing over here in late prophase is our genetic variation, is when we're starting to swap genetic material. So this is where our crossing over happens. And we're still at the 4N. Metaphase 1, so chromosome pairs align and the nuclear membrane breaks down. We're still at that 4N. Anaphase, spindle fibers from the meiotic spindle draw one pair to each end of the cell, and the cell membrane begins to pinch in. Telophase 1, nuclear membrane reforms around the new pairs and 2 haploid daughter cells are created. But each chromosome is still in its replicated state, so it's still in its 2N, which is important. Now we have 2 cells, and both of these 2 cells undergo the second round for meiosis. So for our first round in oogenesis, the first round, we've now created one that's really quite strong because the cytoplasm hasn't divided evenly. This other one is called a polar body, and it's kind of redundant. We can never fertilize that. Whereas in spermatogenesis, we're starting with 1, we're now at 2, and we create 4 sperm. So the process of doing that for this round along here is prophase 2, nuclear envelope breaks down, meiotic spindle is recreated. We're still at our 2N. Metaphase 2, we have our chromosomes which move to a central position. So again, that metaphase, that lock on. Anaphase 2, the centromeres split, separating the sister chromatids and their single chromatids to move to either end of the cell. So the centromeres don't split at our metaphase and anaphase 1, so just check back in that with a checkpoint in a minute. But then we go through to telophase 2 and cytokinesis. The cytoplasm divides and the nuclear membranes reform, and now we have 4 cells with 1 N. So 1, 2, 3 for oogenesis, but 1, 2, 3, 4 for spermatogenesis. So the crossing over is an important part of genetic variation in sexual reproduction. And the recombination occurs between homologous chromosomes. So for me, I have one set of my maternal and one set of my paternal DNA. And at this point, my homologous chromosomes, they're going to cross over. So now one of my chromosomes will have, say, the eye color of my mother, and the other will have the eye color of my father. So I'm no longer displaying dominant and recessive genes, which we'll talk about a little bit further on, within my set of chromosomes. So what is happening is it's crossing over and it's creating new combinations, which is why it's called recombination. The switching of genetic material occurs at the same locus on the chromosome. So there's recombinants of the same gene, so eye color or hair color, between the sets. The point where the crossing over is occurring is called the chiasmata for plural, or chiasma. And it's kind of like a ladder that unfolds, so it allows it to all to cross over. This process creates new combinations of the same genetic material at the locus of the crossover. So this means it's only ever going to cross over eye color with eye color, hair color with hair color. And that's what the chiasma does, is it creates that ladder so that we're crossing over the right sequences. So here is a summary of mitosis versus meiosis. So you should look at this and have this and be able to reproduce this in your study notes. And perhaps you could use a cue card, might have to be a large one, to ask you to draw the events of meiosis. So this is a really important concept, mitosis versus meiosis. And this little summary table down the bottom left is also very important. We're always comparing and contrasting in biology, so you need to have that. So a quick check off. The key events in meiosis that result in the production of gametes from somatic cells, including the significance of crossing over of chromatids between the homologous chromosomes in prophase 1. So in prophase 1, that's where we're crossing over. And we're not dividing the centromere in metaphase 1. Remember, we're pulling back still those pairs in metaphase 1. We split into single chromatids in metaphase 2 and anaphase 2. So to look at sexual reproduction and genetic variation, each gamete has its own combination of alleles. There will always be similarities, but the offspring will always be genetically different from the parents and usually one another. So alleles is something that you should possibly put on your cue cards so that we can understand that there'll be similarities, but offspring will always be genetically different. That's why I look different from my siblings and why you look different for yours, even though you have the same parents. So this genetic variation arises from gametes only having half of the genetic information of their parent cell, and it's randomized. So therefore, which chromosome ends up with which gamete is totally random. When chromosomes pair up during meiosis, crossing over is random, and therefore each combination of genetic material is random. So when we're crossing over, my eye colour from my father and my eye colour from my mother is crossing over. So now it's not one half's definitely from my father and one half's definitely from my mother. It's now, well, one half could be from both my mother and my father because I've got that crossing over happening. And it's occurring in both of my sister chromatids that I have replicated. So meiosis ensures that there's a wide range of genetic combinations occurring between sperm and an egg. And this results in an increased genetic flexibility and increase in the populations. So that increases their opportunity to survive and adapt, most importantly, in evolving environments. So now you can check off how an offspring from two parents has a unique genetic identity, and the key events in meiosis that result in the production of gametes from somatic cells, including the significance of crossing over of chromatids between the homologous chromosomes in prophase 1 and the non-dividing of the centromere in metaphase 1.